And good evening, everyone. I'd like to thank you for inviting me to this opportunity to share my research. I will share my screen here. Does everyone see the screen?
Yep, looks great.
Okay. Let me just pop the window here. It's a bit smaller, maybe. So today we'll talk about bioluminescence. Some of you have seen it up to Oceanside. I heard some of you, speak about it. I will go through a variety of different topics that try to give you the flavor of the different things that are being done in my lab, but also elsewhere. And we'll go from the fundamental research to some of the applications. I will try not to dive too much into the science, but feel free to, of course, ask questions after the fact. So as a biologist, I believe in evolution and that organisms have adapted to where they live. And so if you consider that planet Earth was a pretty, dark and non-colorful space millions of years ago, it has evolved into something very, very pretty and full of color in what we know today. And so if you can visualize that as the squares here being the different environments and the organism living in these environments being the circles, as an organism, you want to match where you live so that you are camouflaged and you are not seen by any predator, or you want to stand out of your environment so that you can tell others that maybe you are dangerous and you want to be seen. That's also the case if you want to be seen because you are looking for a mate, for example. So through evolution, this has evolved into very colorful organisms that have adapted to the color of their environment to show those different sense of do you want to be camouflaged or do you want to be seen? You have experienced that very often, I'm sure. You can see organisms in the tide pools that sometimes you just look 10 times, you don't see anything, and then suddenly say, oh wow, this was there all the time, I never seen it before. Some organisms are able to match their background to be camouflaged, or they can be, of course, very colorful. And if it's colorful, you might probably see, oh, that's dangerous, that's toxic. The same way a ladybug is brightly red because it contains cyanide and it's toxic for you and for any bird that will eat it. Many of those organisms that are very colorful are also potentially toxic, and predators learn about that. So this is just to set the frame of what people usually know and what usually can relate to. Now, if you move into the area where there is no sunlight, well, color doesn't work, right? You cannot see anything. So what animals do is that they produce light for exactly the same function. So animals can produce light in the sense that they will be producing light to be camouflaged, like you can observe here. But they can also produce light, very bright flashes of light so that they can be seen. And those two are similar to what you can observe in the color, the pigmentation that I described earlier in the introduction. And so what is exactly bioluminescence? Bioluminescence is the production of light, visible light, by living organisms. And I say visible because it is associated with an ecological function. You and I produce light, but it's only small photons of light that our nerves produce and that are not to be seen. And so examples of that are usually the firefly, people that come from the Midwest, that's what their reference is. And if you talk to a younger generation, of course, it's E.T. or Avatar. That's most of the time the reference for bioluminescence, because these movies have been inspired by the world of bioluminescence in the ocean. And so that's where you have a protein, basically, that produces light. And so that reaction that produces light, it's a reaction that always depends on oxygen. And based on different cofactors, can produce, an oxidized product and then light. This is the kind of reaction that you see in these glow sticks that you can wear at, parties and New Year's Eve. Those are the ones that inside you have basically a glow stick. You have the luciferin plus the luciferase that can be mixed when you break the glow stick. So for definition, this is very different from what people usually refer to phosphorescence and fluorescence. So phosphorescence, you might have heard the term, oh, phosphorescence C. This is not the correct term. There is no phosphorescence C whatsoever. Phosphorescence, it's a product that is usually non-natural and it is man-made. So phosphorescence is the light that is produced in those glowing stickers that you maybe still have in your room, when you go to sleep at night. It is basically a material that is able to absorb energy from the sunlight and release it slowly afterwards. And so it glows at night. Whereas fluorescence is when you have a blue light that can excite the protein in which you have a transformation of the light into another color of light, usually green, that will be fluorescing. And so when you stop the energy of the fluorescence, you obviously stop the output as well. And so you don't have that delay that you otherwise observe in phosphorescence. So these are the two terms that we have to keep in mind. And we are talking today about bioluminescence, so the animals that make their own light. So as I said, phosphorescence is when you have to excite with blue light, and when you remove the energy, you have still something glowing. And fluorescence is when you excite with the blue light, and then when you remove the blue light, it is basically the fluorescence is gone. And yet fluorescence has been used quite often because many products produce fluorescence. And then it's so visual that also, there has been some biotech push for making things fluoresce because we can see things that otherwise we can using a blue light. CSI and other things like that have used that technology. So bioluminescence is everywhere in every single phyla except the higher plants. And what you observe here, it's a kind of a template of what kind of diversity that you observe in organisms, both in shape and color. As you observe, you have light that can go from yellow to blue, yellow in terrestrial, blue in some bacteria, or yellow-green in terrestrial. So what's going on with this diversity? Well, you have to think that back to the first slide that I showed, that animals produce light so that the light can be seen. And animals that are terrestrial will produce light that is usually yellow, whereas the ones that live in the ocean would produce blue. And that's because when you look at how much light is found in the environment, the organisms that live deep in the ocean are surrounded by blue light, and the blue light travels the furthest in the ocean, whereas on Earth, the yellow light is what travels the furthest. That's, for example, the reason why car lights, your headlights, are usually yellow, so that you can see further away. If you buy this new technology where your car has the LED and it shows blue light, you will actually see much less than what you would see if the lights were yellow. That's just a property of the physics of the atmosphere in terms of propagation of light. So the diversity of color that we observe in bioluminescence is to respond to the match of where the organisms live. If you are living in a deep ocean and you produce yellow light, it won't travel very far and no one will be able to see it. And so vice versa for the organisms that produce the yellow light on Earth. So as I mentioned earlier, bioluminescence has been for a long time, since the 16th century, associated with vision. And actually the first scientists that studied bioluminescence thought that the organs that were producing the light were actually modified eyes. Because if an eye can capture the light and produce a vision of it and an image of it, well, it would be normal that an eye can also project the light out and be doing the reverse function of what the eyes that you and I know about, that we can see. And so in the old days, photophores, that we call them, the organ that produced the light, were seen as eyes that were modified. So vision is always associated with bioluminescence, and that's why in the deep sea organisms have very large eyes, very, very large eyes. And that's because in the deep sea, you want to see the single little amount of light that is being produced, can be a mate or can be a prey or can be a predator. So you need to see that. That's very different from when you go to a cave, a sea cave, where eyesight has been lost. So organisms that live in sea caves usually have developed other senses to go around because there is no bioluminescence ever found in a sea cave or in fresh water, probably because those environments are too dynamic and too young relative to the ocean. And so sea caves are for blind animals, and in the deep sea, even though both of those environments are dark, the deep sea has a lot of bioluminescence, so the eyes are very large. Suddenly, so in this case here, we'll cut the sound. The visual function associated to bioluminescence, associated with bioluminescence, can vary very greatly. And of course, these are interpretations that are anthropogenic, right? We didn't ask a squid, a worm, or something, you know, why do you produce light for? And these functions can be also multiple. They can act in synergy. But what is clear is that some of these bioluminescence occur to like a flashlight, where you find your prey. You basically shine the light around so that you can see where your prey is, and flashlight fish do that very well. But you can also find your mates, which is very typical for fireflies, but also ostracods, which are the fireflies of the sea. They produce certain codes, like a Morse, that will tell, oh, that's a mate of my species. I can swim towards that, or I can fly toward that tree if that's a firefly. Polychaete worms produce glows of little bombs of mucus that will glow so that it can attract the male or the female to swim into that cloud and mate. But the most common one is, of course, the deterring predators. You produce light so that you blind your predator, you know, even temporarily, enough time for you to escape, or you scare away a predator by producing intense flashes of light, which is most of the time what you might have experienced if you were to handle a firefly, for example. And so some of these functions are difficult to assess because working in the deep sea environment where bioluminescence is very common is not easy. You might have a lab at home, at work and everything, but at the end where you do your work is in this kind of environment, is a submarine that is designed to go in the deep sea so that you can see everything. You can collect and suck up the animal using this kind of suction cup here or vacuum cleaner, and then store them in these vessels. And the big problem is, of course, that when we do research in the deep sea, by the time you bring those organisms back to the surface, they are usually dead or dying, and it's very difficult to study deep sea organisms because we cannot keep them under pressure, and they suffer from a shock of temperature, and of course a shock of change of pressure. But it's exciting to do that. You see the little screen here. It's basically whenever the submarine like this goes down, it allows species to hit that screen, and we record the amount of light that is produced. In this particular setting here, this is Michael Latz, my former postdoc advisor, and Eddie Witter, which are two expert in bioluminescence as well. And they were trying to collect samples to put back down in those jars. Now, I told you that screen. How does it look in the screen? So when you have this submarine going in the deep sea, you have things that basically get stuck on that screen, that get bumped, and the mechanical agitation is what makes those organisms produce light. These organisms can be ctenophore, gelatinous organisms, jellyfish, like you can see here, some different fishes as well. And that's to the extent that we can actually observe bioluminescence in the deep sea. We still don't have the technology to go there, hang on for quite some time, so that we can actually see naturally how does the bioluminescence occur, and then what function does it have. So it's not like you can sit under a tree like you could do if you were studying fireflies. Working on deep sea bioluminescence is very, very difficult. And also many cases, these species are unknown, some of them being totally new to the tree of life. So what I showed you was a species that all the species that were basically, pelagic, they were swimming. In my lab, we study a species of deep sea stalk sea anemone. It's Umbellula. So it's about like 50 centimeters, a foot or foot and a half. It lives in the sediment. And what was fascinating about this is that it produces, beautiful bioluminescence. Of course, this organism was brought back, through, a net. We can image them. We can take pictures, beautiful pictures. We can look at various different spectra. But the beauty of it is, of course, how does it produce light? And I teamed up with Carl McBurney, who is a videographer for Nat Geo, who basically filmed this in my lab. So this is the first time bioluminescence of this organism has been filmed into the kinetics. Don't forget, this is a jelly organism. There is technically no nerve associated with it, and you can see how fast this, bioluminescence is being stimulated, not from touching the actual area just under the callus, but by touching just the foot of the organism. So there is a sensing mechanism that allows the stimulation to go along. This is, of course, unknown. We don't know how it happens. We don't know the chemistry of this light production. We don't know much about it, except for what you just saw. This is not even published yet, so you are the first people to watch this, and I hope you enjoyed this setup that we put together. So of course, now if you look at the very deep sea where everything is dark, you go a little bit shallower, which is a few hundred meters still, and it's the dim twilight. So it's dark, but you still can see something. And so in this case, these organisms produce light mostly from their ventral side. And why is that? It is not a random distribution of light. So if you see the lights produced by the fish, is the dotted line here. And that bioluminescence is basically the same color of the environment that the fish lives at. So remember the squares, the circle I showed you at the beginning. You want to match the color so that you disappear, so that you're camouflaged. This is exactly what those fish do. If you look at these fish from underneath as a predator, you actually would not see them if they were producing the same amount of light as their background. And so this fish here that you will see in the movie is able to see the color that this environment is producing, and is producing the bioluminescence that matches exactly the same color. Now, how does the information go from the eye to the photophore is still unknown. It's a black box. This is very common in organisms that live in the deep sea. It's also very common for organisms that do not produce bioluminescence, but can produce light, the same way sharks are white. Because if you are under a shark, which I hope you are not, but if one day you are, you will not see it because it would basically be blending with the background. And so an experiment has been done where we put one of these fish in a tank. You can vary the intensity of the light, and you will see that the fish will follow the intensity of the light that is changed experimentally through this continuous line, and the fish is the dotted line, until, of course, it's too intense and the fish cannot match the intensity. This is shown here in a video that was filmed by the BBC in the late 90s, where
natural ability
they put two fish, one that is producing bioluminescence and one that is not, and they were turning back and forth the light, and you see that both fish are seen, but once the light is turned on, the fish that is bioluminescent is basically almost totally invisible. This is filmed from underneath with the light that is being shined from above. It demonstrates very well that producing bioluminescence can make you invisible for a predator that is under you. Okay, so now let's go back up a little bit further towards the surface. That's where most of my research is done because it's easy access. You have to work at night sometimes, but it's always very exciting and very spooky sometimes, but it's worth it. It's magical as well. Magical is something that you have seen here. This is the red tide that you can observe at the Scripps Pier, sometimes all the way to Oceanside. You can walk on the beach and you can, see all these glowing little particles, which are dinoflagellates. They are little one-cell algae that, when they are agitated, produce bioluminescence. This algae is found everywhere in the ocean, and when it blooms to concentration that are high enough, you can see organisms swimming by and also producing, the shear of the bioluminescence. With here, Michael Latz, my former postdoc advisor, was studying how a dolphin swims through this, and would observe that the shape of the nose of a dolphin is such that it does not create bioluminescence, whereas the rest of the body does. And this is because the nose is able to open the water in such a very smooth way that it provides the least amount of shear for the dolphin to swim through, which is basically based on this inspiration that boats now have this big nose around, the cargo ships, so that it can just break open the water in a very evenly way. So these are the organisms that produce the bioluminescence. They are very, very small. They are just, 30, 40, 60 micron. In the audience here, we have a few experts that are known to study them. This is the one that produces the bioluminescence here. It's one cell, and it produces bioluminescence from the different little vesicles called scintillons. That's where the bioluminescence is produced. And it's always produced in response to a mechanical agitation. So why do they produce that? Well, if you are one little cell like this, and you have all your millions of brothers and sisters, so you are almost all identical. And if you are being exposed to a predator that is an ostracod here, a copepod that can eat you, but there is also a fish here that can eat this primary predator, the copepod. So basically, what it happens is that if the copepod eats the one-cell algae, it will start glowing. Then the glowing copepod will become the prey to the fish. And so that burglar alarm mechanism allows one-cell algae to sacrifice itself for the other millions of cells minus one to stay alive by sacrificing one of the one of their member. And so that burglar alarm is pretty fascinating, and it works very well. I've been in swimming through these glowing waters. If you shake hands, you can have actually fish swimming fast, and you of course believing that there is something big there that they can feed on. So be careful when you do that. Of course, the beauty is there. So we can keep those samples in flasks. We can grow them in the lab. You can shake them. You can see the light. You can look at them under the microscope. It's a beautiful system that you can study and that you can recreate in the environment and have a material that you can then make glow at will, which of course invite to art. So there is a lot of applications for this particular organisms that produce bioluminescence. This is biopop, now called Pop Bio. Actually, they make structures that glow in the dark, carpets with living cells in them. You can dance on them. You can make all the shapes you can imagine. They were very famous at some point for what they call the dino pet. But also bioluminescence is used as an extract of chemistry. So the chemicals that are found in those organisms do chemiluminescence. So if you remove the organisms like the glow stick, you can actually make glowing liquid. And this glowing liquid obviously is the source of inspiration for light that you can see at home. Maybe one day will be lit up at home or at camping sites with this biological light. The ones in the bucket under show the details of, again, of the dinoflagellates. And this light can be very bright. You can actually lit up an area, but of course it's so related to the shear that you constantly need to have the water move. And of course, at the end, of course, the organism also get tired and fatigued. And so this is still a work in progress if one day we were to use bioluminescence from these organisms as a source of light. It is also used as a source of art. This is the Infinity Cube, where this was an art from Yvonne Koo and Mike. They were filming bioluminescence of cells that were going through the field of view, putting that into a cube made of mirrors. And you would sit there and be surrounded by these flashes of light. It was, it's an incredible exhibit that was at the Birch Aquarium a few years ago. So aside from these organisms that produce light, I will just go through very quick other organisms that we study in my lab. You can see sometimes brittle stars in the ocean, in the tide pool. Sometimes not all of them produce bioluminescence, but this one in particular, Ophiopsila californica, produces bioluminescence. They live deep in the sand. They have their arm sticking out. And when you actually stimulate the, try to grab these organisms, they will produce an intense amount of light that will stun a crab and scare a crab away. So the crab is ready to eat it, and it's like, oh, what is that? No, I'm not eating this. We can create that same effect in the lab, of course, like we can do here. You can stimulate the light with different chemicals. But in the field, what is clear is that the crab, he got scared away enough that he actually is not feeding on any of those brittle stars and decides to just skip that lunch for now and then prey on something else. And this is an image that we create in the lab by stimulation under a chemical substance. Now, the bioluminescence of the brittle star is very interesting to us because it is controlled by a nervous system. And that brittle star in particular has the same nervous system that you and I have. And so we can use this brittle star as a proxy to understand what are the different chemicals that affects the transduction of a nervous influx. So instead of doing that on organisms like humans or rats or anything like that, we can try to start looking at the effect of drugs on these organisms and look at use the light as a proxy for the effect. We also study a worm. As you have seen until right now, the light was always produced as flashes, right? In this worm, for example, Chaetopterus variopedatus, we were able to measure light all the way to 16, 17 hours, which is extremely long when the light is produced through a mucus, so it's secreted. How does it work to produce light, which is very energetically demanding, to last so long? And so, in a nutshell, this worm produces a very, very thick mucus that we study, we characterize very well. And what's interesting is that that mucus contains a lot of ferritin. And the ferritin seems to be the battery that allows the light to be produced. That ferritin is very similar to the human ferritin, but it's much better. So human ferritin is here in red. The worm ferritin have been optimized to react even faster in reaction to iron, to the point where we are considering using this ferritin to detoxify against the iron toxicity in some patients, because the worm ferritin is much better at capturing iron than the human ferritin is. All that because it probably uses that to control the bioluminescence. Why does it produce the bioluminescence in a sticky mucus? Again, this is hypothetical, but this is the most logical answer that we have. If you have a fish that comes and bother the worm will produce this very sticky, glowing mucus that will go all around the fish. The fish will then have a tag along its body that will be sticking, that will be glowing for 17 hours, 24 hours. And of course, here comes the shark that will see the glowing prey, and then here goes your fish, bye-bye. So that's, again, a way to defend itself, but also in the long term. So your fish attack you for 1 minute and will be tagged maybe for 24 hours and will be eaten and will not attack you again. Second to last example, we study also Hemia brasiliana, which is, as I call it, the high-tech snail. This snail produces light, and there is another snail that produces light, and it produces light from the foot. And so when we discovered this particular snail, we thought that that snail would produce the same kind of light from the foot, which is, you know, logical. So the snail would just go around, and then the light, it would produce light, it would leave a trail like it was described before in another species. And then, of course, you have all played with snails before. If you tap on them, they go under, right? And so you expect the lights to not be visible because then the light, the foot, will be under the shell. So that's assuming that the light production is originating from the foot of the snail. So we did that experiment where we put the snail into a petri dish in an instrument called a luminometer where we can record light. And we have individuals here of snails. They produce flashes of light. They are alone, right? There are just 3 of them. They just are chatting, maybe, who knows what they are doing. And this is the background of light. So we know now that they produce light spontaneously, just by going around. They probably are, you know, cruising through the petri dish. Now we open the luminometer and we put a little starving crab in it. So the crab would definitely try to eat the snails. And what we expect is those blue, red, and green light to drop where the black line is, because we assume, of course, that the bioluminescence is on the foot, and that upon predation, the snail will go under the shell, and the shell will then, you know, block the light. As you have seen, the shell is opaque and is yellow. And of course, nature is beautiful. What you observe is totally the opposite. When you expose the snail to a predator, it produces, you know, 1,000, 10,000 times more flashes of light. How does that work? In that particular snail, the light production is produced from under the shell, not outside the shell, to the point where the shell appears as a light diffuser. It basically, this area here where the tissue is, it's barely the size of 1 millimeter square, and the entire shell is able to glow based on that just one single area. So it's amplifying the spatial distribution of the light. How does it do that? It does it in a way that is even better than what a commercially available light diffuser would be. Those are the little pieces of plastic that you observe, that you have on your flashlight or on your ceiling or in your lights at home. Those are light diffusers. The snail is able to diffuse light even better, about 10 times better. It does it because the shell is organized with little microfibers, optical microfibers, that are able to capture and spread all those photons of light to the biggest space possible. It's made of aragonite, which is the same material of your teeth, but the aragonite is made in those little optical nanofibers or microfibers. So very sophisticated shell that is able to therefore capture this light and spread it so well that now it can be used as a light diffuser for non-toxic material, obviously, maybe for solar panels and other things like that. Okay, so back to Earth. This is the last, few example that I want to show you about how big we can think. So if you play with a firefly, you can see that the light is produced here at the bottom, and the light is produced internally, right? It's inside the body. So the question is like, how does it transmit the light from deep inside the body to the outside? Because you still have a skin and a layer of chitin to go across. And so when you look at the area where the light comes from, the light actually is scattered by the chitin, but the chitin is organized like the tiles on your roof. So it's not as one single plane unit, because if you have a plane unit, if you produce light, it's being scattered away from that plane. It's basically will reflect off the surface. So if you look at a plane surface, only 20% of the light will go through. If you have jagged scales, actually you increase that by 10%, which is amazing that just by this little change in engineering of a surface, you can actually increase dramatically what we call the extractability of light. And why is this important? Because your lights that you use for Christmas only let 3.7% of the light from inside to the outside. So what you see from those little bulbs is only 3.7% of what the actual amount of light is inside. So we are losing a huge amount of light. But if you were to pattern the surface of those little lights, those little bulbs, with the inspiration from the firefly, you can already almost double that and make your LEDs much brighter. So that's one of the applications, for example. And the last one is that fireflies produce light with a chemical reaction that is the well best known in the world of bioluminescence. And it produces light with ATP, adenosine triphosphate, which is basically a molecule that is found in every single living organism. It's the source of energy, is what every single cell uses. So because of that, scientists have taken the extracts of firefly and put them on Mars in the rover. So the rover now is taking samples of the soil and mixes the soil with the extract of the firefly. And if there is a flash of light, that means that something that is similar to ATP, assuming that life on Mars would have used ATP as well as a conduit of energy, but it is used on Mars to detect any kind of possible life. And so I think it's fascinating that these applications that I mentioned to you can go anywhere from just looking at the biomedical application to visualize some things to something that is used by NASA on Mars. And with this, I'd like to thank you again. I had a lot of ways to thank. I don't have time to thank all the undergraduates and graduate volunteers that work in my lab on this topic. But it's, I'm sure they, hopefully they all enjoyed my work on this magical topic, and I will take any question. Thank you.
Wow, that's just fascinating. Thank you for sharing that. One question we had is, are the dinoflagellates that produce the bioluminescence, that we see in the waves sometimes, are they always in the water? And if so, why don't we see them all the time?
So the dinoflagellates are always present in the water, but they occur in very small concentrations. And then sometimes you have conditions that are favorable for one species to grow better and more than the others. And if it's a species that produces bioluminescence, you will have a bloom, and that will create this very massive amount of cells, and you will see a lot of light. If you go swim when there is no red tide, sometimes you can shake your hands and you will see a little speck of light here and there. So those cells are always there. They are just not very concentrated. Now, scientists have always tried to predict, when does that happen, right? And it's unpredictable so far. What we only know is that you need to have certain conditions. You need to have a lot of nutrients, because of course you need to double up, you need to grow, you need to bloom. And also, you also need to have sunlight and warmth. So you need warm water. And so it happens here in San Diego mostly when this happens, which is the time between seasons. So when you go from winter to summer, and that you start having winds that would push the deep water, very cold and rich in nutrients, to the surface, and you have a very hot day following that very rich water that warms up might lead to a bloom, and that actually might lead to bioluminescence. The same way, reverse, in the fall, you can have very warm water that will have cold water coming up from the deep that will be rich in nutrients, and you will have the same phenomenon. So we know that this is driving the blooms, but we don't know if it will last 24 hours, if it will last 24 days. We had it last for many months in the past, because it's extremely dynamic and depending on winds and currents, but also on the stratification of the water in terms of the the distribution of nutrients.
Okay, great. We had another question. Do any terrestrial snails have bioluminescence?
No, none of the snails that are bioluminescent, that are terrestrial, are bioluminescent. The other snail that I mentioned, people confuse it sometimes as it could be called terrestrial, but it's actually living in a stream. It's actually always living in a very wet environment in New Zealand. That's the only other case where this is known. So it could be called terrestrial, but it's freshwater to some extent.
Okay. I don't see any other questions in the chat. Does anyone want to unmute themselves and ask a question?
In the ocean, is any of the bioluminescence ever used to attract a mate, for example, rather than just to deter a predator?
Yes, we believe that's the case. If you remember in the movie, Finding Nemo, where they have the anglerfish that has this this glowing bulb in front of the head, that is used to attract a prey. But it is also there is indication that the some organisms can use that to attract their or at least to indicate potential mates that they are there. Because these organisms have also markings on their body that is not necessarily the use for attracting prey or for deterring predators. Now, some of the fish that I showed are also known because their sexual partner might be a very miniature, very small mate that, I don't want to give details, but that basically screwed itself inside the body of the bigger fish, which is the mom. So the male will be a tiny little organism, little fish that will anchor itself inside the mom and will stay there. And that's how it finds its host. Does it use the light of the female to see that it's the right species? It's possible, we don't know. But the male itself does not produce bioluminescence. It's this very tiny little fish that will just have the function to release its gamete.
Interesting.
But not all the fish are like that, right? Some fish, for sure, we know have patterns that can, maybe be species specific. There are some sharks that produce bioluminescence that we still don't know clearly why they produce that. They have only certain parts of their body that produce light. So your guess is as good as mine, except that we rely on observations that sometimes are from divers or people in submarines, and it is very hard to, because you and I see the light maybe very differently. We know that the vision of these organisms might not necessarily be what you and I see. They might have a channel vision, they might see a different color, and they might see things with different contrast. And so what we observe, us as humans, as something that might provide camouflage, might also provide something else than the camouflage. So these functions are, is what we have to work with, and we can only add observations and tests in the lab to try to say which one is more dominant than the other, or maybe all of them co-occur in one single species.
Right. Thank you.
Do you have any other questions? I'm not seeing anything else coming up. So wanted to thank you so much for sharing with us. It was a really interesting topic tonight. And we learned a lot, and we're getting a lot of thank yous. Fascinating topic.
Great, thank you.
We appreciate you spending time with us tonight.
Have a nice glowing evening.
And we'll look forward to the next red tide so we can see the waves glowing, and now we'll understand a little bit more about how it works.
Yeah, we had a red tide recently, but it was not a species that produced bioluminescence. It's another guy that took the lead. And so it's always a competition between species. So the ocean was red, but it was not producing light.
Okay.
Interesting. Okay, thank you.
Yeah.
Great. Well, thank you for joining us tonight. We appreciate your time. And I guess we'll wrap it up. It's a little after 8, and thank you everyone for joining us tonight.
Okay, thank you, Joni.
Thank you.
Thank you, everyone.